Hi everyone, this is Dr. Fox. I hope all is going well. Let's talk about the leukocytes. In this video, we'll discuss the types, development, functions, locations, and relative abundances of the leukocytes. First of all, leukocytes are also known as white blood cells, and they are predominantly important for defense of the individual. So they are going to encounter pathogens and have different strategies for helping defeat them. They are very easily imaged with light microscopy, and they appear lighter than the surrounding erythrocytes or red blood cells, oftentimes also appearing slightly blue or even purple. They will circulate throughout the blood, and some of them are going to move into the tissues where they will ultimately live or function. We can distinguish two major classifications of white blood cells: granular versus agranular. Granular leukocytes, through light microscopy, have granules within them. Oftentimes, these granules are going to contain substances that are enzymatic or destructive. They help with the function of the leukocyte, whereas agranular leukocytes do not contain these granules and they are not visible under light microscopy. Some of the functions of leukocytes include phagocytosis. So phago means to feed, cytosis on cells, so this is cell eating. They can engulf pathogens, cells, or other structures and then destroy them. There's also chemotaxis, which means that they can be drawn towards a chemical signal, and oftentimes this chemical signal is where they are drawn and then they will exit the bloodstream into the surrounding tissues. And emigration, also sometimes known as diapetesis, is when the leukocyte, which has been drawn to a particular area through chemotaxis, then leaves the cardiovascular system and enters into the surrounding tissues. So we'll see that the leukocytes have different abilities and different capabilities depending on what they are. So their development is fairly straightforward. All come from pluripotent stem cells. And then from there, pluripotent stem cells may either become myeloid or lymphoid stem cells. Myeloid stem cells become a variety of cells, so your erythrocytes and thrombocytes, which are not of the leukocytes. Within the leukocytes, all of the granular leukocytes come from myeloid stem cells. One of the agranular leukocytes comes from myeloid stem cells, and those are the monocytes, which then become macrophages within the tissues. And then the agranular, the remainder of the agranular leukocytes come from lymphoid stem cells. So these are lymphocytes. So T cells or T lymphocytes, natural killer cells, and B cells or B lymphocytes, and their derivatives, plasma cells. So there are a couple of different developmental pathways to arrive at leukocytes. So here, let's start with our survey of the leukocytes with the granular leukocytes and with the most abundant granular leukocyte, neutrophils. Neutrophils are the most abundant leukocyte, and they are typically the first on the scene of an infection. Some folks will refer to them as polymorphonuclear leukocytes, or polys for short. Here is an example of a neutrophil, and we can see that it has several different portions or lobes to its nucleus. Now, also important to understand here is that in order to visualize tissues, histologists will stain those tissues, and it's hematoxylin and eosin, or H&E staining. And so hematoxylin dye is usually absorbed by nuclei and differentially absorbed by some leukocytes, whereas eosin is more absorbed by cytoplasm, and you can see eosin dye absorbed by some leukocytes, but it's most readily absorbed by erythrocytes, and so they are going to appear more pink or red. Now, neutrophils don't last very long. Typically, their entire lifespan is less than a week, but they kind of do it all. So they are phagocytic and chemotaxic. They move throughout the blood, and they will leave the blood or emigrate into the tissues. Now, when they encounter a pathogen, especially one that they eat through phagocytosis, they're also capable of degranulation. So they are granular leukocytes, and they contain a variety of different substances, lysozymes, which are bactericidal, and their granules are oftentimes non-specific granules, which are a cocktail of proteases and acids and other things that, when combined with what is brought into the neutrophil through phagocytosis, is going to destroy it, like a little mini cellular grenade. Oftentimes, one of the signs of an active infection might be purulence, colloquially called pus. That purulence is a collection of dead neutrophils. So that is what is being drained from an infected wound. Neutrophils are also quite abundant. They comprise about 60% of the total white blood count, which is fairly impressive. Another granular leukocyte is an eosinophil. Eosinophils typically appear a little more pink than your average leukocyte. They live for about 2 weeks. They have limited ability of phagocytosis, but they do have chemotaxic ability, so they can be drawn to chemical signals. They're found in the blood, but also they can leave the blood through emigration into surrounding tissues, and typically eosinophils are found in mucosae. When they degranulate, they typically degranulate enzymes. And so a couple of substances, the major ones, not all, eosinophils can degranulate leukotrienes. Leukotrienes are an important part of an allergic response, and they tend to exacerbate allergic responses. Kind of coincidentally, they also can degranulate histaminidase. So histamine is a vasodilator, and histaminidase can actually break down histamine and therefore slow allergic reactions. And then the other major element that can be degranulated by eosinophils is major basic protein. Major basic protein is a very powerful destroyer of various endoparasites. So schistosomes, for instance, can be killed with major basic protein. And eosinophils are not very abundant. They comprise about 3% of a whole white blood count. And then the least abundant leukocyte are the basophils. Basophils get their name because they readily absorb hematoxylin, and they appear very blue or purple. So that is the basophil. That is another leukocyte. So you can see the difference there in the appearance, and oftentimes the granules of basophils may even partially obscure the nucleus. Basophils, unlike many other granular leukocytes, have very long lifespans, so on the order of magnitude of 1 to 2 years. So other granular leukocytes are maybe living for weeks. Basophils live for years. They are predominantly found circulating in the blood. They're very, very rarely found in the tissues, although they do have chemotaxic ability, but not phagocytic ability. When they degranulate substances that encourage vasodilation, like histamine and heparin, etc. And one of the more interesting things about basophils is that when they're activated, they stimulate the formation of a particular type of antibody called IgE that attaches to mast cells. So mast cells will oftentimes secrete histamine and encourage allergic reactions. And this is the basis behind allergic reactions that lead to anaphylaxis. So think of food allergies. If someone has an allergy to peanuts or nuts or shellfish, oftentimes it's the result of basophils putting IgE antibodies on mast cells that that are specific for antigens found in those substances. So these are fantastic amplifiers of allergic responses, but they also can help battle ectoparasites, things like fleas and ticks. But again, they are not terribly abundant, coming in at less than 1% of a whole white blood count. Now let's take a look at some of the agranular leukocytes, so monocytes. Monocytes have a very short lifespan. They exist for about 3 days in the blood, and then as they emigrate into tissues, they go through a process of maturation, some of them becoming macrophages and some of them becoming dendritic cells. So macrophages live throughout tissues where they can exist for months. Two various types of macrophages are fixed, where they stay in one place, or wandering macrophages, which can move throughout tissues. Here's an example of a monocyte, and typically they have very robust kind of U-shaped nuclei, but you'll notice that there aren't any granules in there, so it's a very clear agranular leukocyte. Dendritic cells get their name because as they mature, they have larger sort of pseudopodium or appendages that look like dendrites. These are important because they become antigen-presenting cells, or APCs. And antigen-presenting cells are going to take little bits of destroyed pathogens and present them to lymphocytes so that they can begin to recognize them and become activated for an immune response. So monocytes and their derivatives are very important, not just for innate immunity, but therefore also for adaptive immunity as well. And they're about 10% of all white blood cells. Then finally, we have a rather large class of agranular leukocytes called the lymphocytes. Lymphocytes can be found in the blood, the lymphatic system, as well as in the tissues, and they are the basis of acquired immunity. And there are two general categories here that are doing the heavy lifting: the T lymphocytes, or T cells, and the B lymphocytes, or B cells. T cells come in a variety of different types. There are T helper cells. T helper cells are going to be stimulators of other lymphocytes, such as cytotoxic T cells, which can destroy pathogens. There are also T suppressor cells, or Tregs cells, which are good at suppressing autoimmune responses. And then B cells, or B lymphocytes, once they become activated, become plasma cells, typically in tissues, where the plasma cells are the cells that are secreting antibodies for humoral immunity. There's also a class of lymphocyte that is important for innate immunity, and these are called natural killer cells, which behave much the same way as cytotoxic T cells, but require no previous exposure. Lymphocytes are never, ever phagocytic. And when we look at a blood smear, we can categorize them as either small, which typically the abundance of them, up to about 90% of them, are small, or large. Large lymphocytes are active lymphocytes. So small lymphocytes become large lymphocytes when they encounter an antigen and then mature into a lymphocyte which is capable of mounting an immune response. And lymphocytes as a category are fairly abundant, coming in at about 25% or a quarter of all leukocytes. So as part of blood testing, a physician or healthcare professional might order a differential white blood count, and this could give different information depending on specifically what white blood cells are elevated. But generally, and I don't expect you to read this chart in depth, but we have two conditions. There's leukocytosis, which is an elevated white blood count. This is typically when in a blood sample, white blood cells account for greater than 11,000 cells per microliter. And then there's leukopenia, and that is when white blood cell count is very low, on the order of magnitude of below 4,000 cells per microliter. And you can apply this to any particular white blood cell. So you can have neutrocytosis, neutropenia, monocytosis, monopenia, eosinocytosis, eosinopenia, basocytosis, basopenia, depending on the individual white blood cell. So let's evaluate our knowledge. Which leukocyte would you presume to be the most abundant in a blood smear? Basophils, pretty rare. Eosinophils, pretty rare. Lymphocytes, fairly common, coming in at about 25%. So maybe monocytes, also pretty common, maybe about 10%, but overabundance is the neutrophils. They are the number one leukocyte that one can find in a typical blood smear. Thank you so much for your time, and I look forward to talking to you in the next video. Take care.